This material is made available to you by or on behalf of the University of Melbourne under Section 113P of the Copyright Act 1968. It may be subject to copyright. For more information, visit the University Copyright website.
We both came over to Australia, back to Australia for Dr. Sharpe. So I'm really excited to see her and catch up with her on this side of the world. Mel recently moved her research group from the Department of Psychology at UCLA to the University of Sydney. She obtained her Ph.D. in Sydney as well at the University of New South Wales and completed her postdoctoral training at the U.S. National Institute on Drug Abuse, NIDA, and as well as Princeton University. And she was supported there by an NHMRC C.J. Martin Biomedical Fellowship. Her research group investigates neural circuits involved in reinforcement learning, and she uses a suite of different techniques, including optogenetics and fiber photometry. And she really seeks to understand how dopamine and hypothalamic circuits might be changed in psychological disorders like schizophrenia and addiction. So she's going to talk to us about some of that work today. We're really excited and lucky to have Dr. Sharpe with us. Her work's been supported by multiple R01 grants from the U.S. National Institute of Drug Abuse, as well as an R21 from the U.S. National Institute of Mental Health. And for those of you in the room who aren't familiar with the R scheme in the U.S., the R01 is basically the big honcho, so those are very hard to get and exciting. She's also received a Young Investigator Award from the Brain Behavior Research Foundation, and an NSF Career Award, which is analogous to the ARC Future Fellow that we have here. She serves as editor for multiple peer-reviewed scholarly journals, including her current position at the Neurobiology of Learning and Memory. And she's not only an inspiring woman in science, but she started the UCLA Women in Neuroscience group and is an outspoken advocate for women in STEM. So I'm really proud to be presenting her to the hub today. So today Mel will be sharing her work identifying the function of the lateral hypothalamus in reinforcement learning. This is a significant advance in understanding of hypothalamic function, which has been historically restricted to things like maintaining homeostasis, feeding behaviors, and other innate behaviors. So we're going to hear about its role in learning today. So please join me in welcoming Dr. Sharpe. We'll have time for questions at the end. If you don't get to ask your question in this forum, please do grab her and speak with her during the lunch as well. So thank you, Melissa.
Thanks, Sarah, for that awesome bio. It was really lovely. I appreciate it. So I'm really excited to be here in a kind of cognitive neuroscience space so I can go really learning on this. Stop me at any point if there's anything you don't understand, but I'm pretty sure you guys are going to be familiar with the sorts of tasks that I'm talking about. And essentially, as Sarah was saying, what I'm going to talk about today is my kind of secret passion project, which is the role of the lateral hypothalamus in learning. And previously, this area of the brain is thought to be really important for feeding. So the idea is, if I was to turn on your lateral hypothalamus right now, you'd start voraciously eating anything in front of you and maybe even gnawing your desk or something like that. But as an associative learning theorist who thinks about feeding as something that is learned, to me it seemed really obvious that this area of the brain might be important for critical aspects of learning that we hadn't uncovered yet. And indeed, the work that we've been doing across the best part of a decade is now showing that I think this nucleus is a really important driver of reinforcement learning in a way that I think is clinically relevant. And so I'm excited to be here with a group that does a lot of work in humans, because I am begging people in human research to have a look at this area. People are not looking at it, and I think it's really important, and we're missing out on a critical piece of the puzzle for psychological disorders. So before I start talking about the work, of course, I want to recognize and pay my respect to the elders and communities, past, present, and emerging, of the lands that the University of Sydney's campuses stand on, and of course in Melbourne, that is the Wurundjeri people. So recently, we published a paper in Ticks, which was essentially making an argument that the lateral hypothalamus really is a key driver of learning. This was the first thing I wrote when I came to Australia. It was a joy just to write what I've been thinking about for 10 years. I was kind of sad when I submitted it to the editors. And basically, what I was trying to argue is that the lateral hypothalamus is very specifically biasing learning and behavior towards outcomes in our environment that are important. So that might be food rewards, it might be drugs, it might be social interaction, and also even things like pain and shock. So we have a whole project that looks at the role of lateral hypothalamus in the formation of fear memories. And so today, what I really want to do is to try to give you a kind of overarching idea of what the lateral hypothalamus is doing in learning and why it's important to think about as a critical driver of learning. And so in the first part of the talk, I want to talk about the really key data that led to the theory that we're now trying to advocate towards to argue that lateral hypothalamus is important for biasing learning towards important predictors and close predictors of outcomes. And then I'll talk about some of our really new data that's unpublished, it's not in a preprint, anything like that, and we're recording in lateral hypothalamus during learning, just learning about cues and rewards, after rats have had experience with methamphetamine self-administration. And essentially what this is showing is that the circuit seems to be upregulated following exposure to drugs of abuse that seem to produce reinforcement learning changes that are consistent with what we see in people with addiction. And then in the last part of the talk, I'm going to talk about some really early work, which is trying to develop a translational task in rats to be able to really formally test our theory in a way that lends to computational modeling. And for those of you that are familiar, we're trying to adapt and change a little bit the two-step task from Nathaniel Daw. And so we've been working with him and Marcella Matar at NYU to develop this. And I'm really interested to get your feedback on this project. So as I mentioned, we typically think of the lateral hypothalamus as being important for feeding. And I have never really worked in this area before, but we were doing a journal club in my postdoc lab at NIDA where we were looking at this really amazing paper from Garrett Stuber and colleagues. And they were using all these fandangled new techniques like calcium imaging to look at how GABA neurons in the lateral hypothalamus are important for feeding. So we specifically think now that it's the GABAergic neurons in lateral hypothalamus that drive the effects we see in feeding. And here's the one experiment that they did with optogenetics that really caught my eye and made me interested in this area. And so essentially what they're doing is to put a Cre-inducible channel rhodopsin into GABAergic neurons in lateral hypothalamus. It's the VGAP mouse. So all that means is they're able to stimulate GABA neurons in lateral hypothalamus when they want to using optogenetics. And this is the task that they're going to use. So here is just an open field arena, and you can see these are heat maps of where the mice are moving to. There's an area of this arena that has a lot of food in it, and there's also what they're going to call a no-food zone that they're going to compare the amount of time that they spend in that particular area. And you can see before you start stimulating GABAergic neurons, animals are just walking around and they're not spending more time in the food zone, and that's because they're sated, they're not hungry. And so they know where that food is, they know the area of the arena that's paired with that food, but they also know when they're not hungry that that food isn't rewarding, so they're not going over there to consume it. But when you start stimulating these neurons, so you deliver light into the brain that activate GABAergic neurons in the lateral hypothalamus, they go straight away over to that food zone and they start consuming a whole bunch of food. And this really is online control, because if you stop doing that, they immediately stop consuming the food, and they even go into this kind of opposite end of the arena. And I think about it as, you know, they're kind of gorged when they're not even hungry, and now they're like, oh gosh, like I'm going to go over here, you know. And so this is a really interesting effect, and it's a really beautiful paper. But for me, it made me think of how this region might be important for learning. For example, these animals know where the food is. They know it's rewarding when they're hungry and not when they're sated. And maybe what you're doing when you're stimulating these neuronal ensembles is to generally increase representation of those learned associations around food to drive behavior towards that food. And so the first idea and the first thing that we tested was just really to see if we could implicate these neurons specifically in associative learning about cues and rewards. And so we ran a really, really simple experiment, which was to teach rats that one tone leads to food and another tone leads to nothing. And what we can do using optogenetics is to selectively inhibit the GABAergic neurons in the lateral hypothalamus during presentations of these tones and not during presentation of the food. Importantly, we're inhibiting now because we want to look at the necessity for learning. The last experiment I showed you was stimulating to generate food-directed behavior. And importantly, we just do this during the tone and not the food because we want to look at their ability to associate these two things together without disrupting the actual food-directed behavior itself. So these rats are in an operant chamber. The food will come into the food port. They can hear, see, and smell that food and go over, right? And so hopefully what we're doing is to not disrupt that behavior and instead just looking at their ability to associate the tone with the arrival of food, which will be indicated by how much they go into the food port when those tones are presented prior to food delivery. We have a little 1 second gap here just to make sure that we're not doing anything to the circuit when that food is being delivered, so it really is back to normal and functioning. So here's responding at the end of training. Here's our EYFP group. So in all the experiments I'll show you today, the EYFP group is our control group. They don't have the active virus in the brain that allows us to inhibit these GABAergic neurons in lateral hypothalamus. And the NPHR group are going to be the group where we're inhibiting with optogenetics these neurons. And so what you can see at the end of learning is these animals show high responding in the food port during presentation of tone 1 that's associated with food and less responding to tone 2. These tones sound kind of similar, so you do get generalization of learning, but they're pretty good at noticing the difference and using tone 1 to predict food. And then what you see when we've inactivated GABAergic neurons in the lateral hypothalamus is we significantly reduce how much they're using those tones to predict the arrival of food, and it's by about 50%. And given that we're not affecting all of the neurons, this is a pretty big effect. You can imagine you'd be affecting about 70% of the neurons with our optogenetic techniques. So this is consistent with an idea that lateral hypothalamus neurons are really important for associating the tone and the food together. But of course, we also want to make sure that they're responding normally during food delivery itself, and that's not why we're seeing an impact on learning about the tone. And you can see that after the tones have been presented, they're really good at going straight to the food port and consuming that food. There's no difference in latencies or amount of time that's spent in that food port. And then the final thing we want to do, because we're good associative learning theorists, is to present the two tones, tone 1 and tone 2, without inhibiting the neurons and just see what they learn. Because of course, when you're actively inhibiting the neurons with optogenetics, you could be changing motivation or attention in a transient way that's not actually reflecting what they've learned. And so when we present those two tones, we see that we maintain the deficit in our experimental group, the NPHR group here, which indicates that we did have a lasting effect on the ability of them to learn that the cue predicts the food. So essentially, this was the first demonstration that the lateral hypothalamus is actually involved in learning about the predictors of food over and above its role in feeding. And it's a really simple experiment, but I think it's really important because what it suggested is that this region is contributing to learning, and so it could be doing all this other cool stuff too. And that's essentially what we were interested in understanding. And so one of the questions that we had following up from these initial studies were to ask what would happen if we were to inhibit lateral hypothalamus as rats were learning about neutral cues and the association between two neutral events. Would the lateral hypothalamus also be important for associating those two neutral cues together in the way it was important for learning about the cue that predicts food? And to do that, we use sensory preconditioning. And so this is something that's seen across species, so you might have seen it before in humans. And how we do this in the rats is essentially to first teach them that there's a relationship between two pairs of auditory cues. So for the rats, we present a 10-second auditory cue, A1, and that's followed 1 second later by A2. And on other trials, we present B1, and that's followed 1 second later by B2. So they're learning that there's a relationship between A1 and A2 and B1 and B2. And once we've taught them this, we take them into the same chambers on a different day, and we're going to teach them that one of those cues, A2 here, leads to arrival of food, and B2 does not. And so what we come to then is the critical probe test, where we're trying to understand what they learned about the structure of these associates in the first phase. And so the idea is, if they associated A1 together with A2, they should respond as though they expect food to be delivered when A1 is presented and not when B1 is presented, because A1 is associated with A2 and A2 leads to food, so A1 might lead to food as well. And here's the data from our control group here. You see a very modest sensory preconditioning effect. So you can see that they increase responding to A1 relative to B1. Turns out having these 1-second gaps, which we don't usually have, so that we can selectively inactivate GABAergic neurons just during A1, actually reduces the sensory preconditioning effect, which is kind of interesting in and of itself. And when we inactivate GABAergic neurons in the lateral hypothalamus during A1, and we're not impacting them at all on the subsequent phases, this allowed us to see a significant increase in sensory preconditioning. This was the opposite to what we expected. And what it suggests is that actually inactivating the lateral hypothalamus during A1 actually enhanced the development of the A1-A2 association, which meant that we saw more food-directed behavior to A1 in that critical probe test. Does that make sense? This was confusing at first. We were like, what is this? This is crazy. But then it kind of makes sense, because if you think about sensory preconditioning, and particularly the first phase, there's a couple of things that are going on, right? Okay, these animals are in a chamber. There's some auditory cues that are being paired together. They should learn about that. They're reliably related, but they shouldn't learn too much about it. There's no food, there's no shock, there's nothing that's important to the animal. And so what I think we're revealing here is that the lateral hypothalamus is part of the circuit that's going, hey, don't learn too much about this. There's nothing important that's happening right now. And so when you take out that influence by inhibiting the neurons, you're letting the other systems kind of learn about these auditory relationships in a way that they shouldn't. So what we think this shows is that the lateral hypothalamus is opposing learning about neutral information and neutral cues. Does that make sense? Sometimes it's a little complicated to get your head around. It was for me when we first found these data. And so we have 2 more experiments that I'm going to show you today that support an idea that what the lateral hypothalamus is doing is to oppose the development of learning about irrelevant information, neutral information, or information that is distal to those really proximal reward cues. And so the second experiment that we wanted to run here was to understand if we made one of those neutral cues motivationally significant, would suddenly the lateral hypothalamus care about them, and suddenly it would want to learn about the relationships between the neutral cues. And so to do that, we use second order conditioning, which is exactly the same as sensory preconditioning, but you just move around the order of phase 1 and phase 2. So the idea is you first teach them that A1 leads to food, B2 leads to nothing, and then you teach them the relationship between A1, A2, and B1, B2. So A2 is motivationally significant here, and other areas of the brain that don't care about neutral cues and sensory preconditioning actually come on board to now care about this relationship. So we thought maybe lateral hypothalamus is the same. And you expect the same effect in the probe test, which is more responding to A1 relative to B1. But what we're going to do is to inactivate GABAergic neurons in lateral hypothalamus during A1 again, and see if we disrupt that learning now that A2 is motivationally significant and A1 is a distal predictor of reward. So in our control group, again, we see a modest second order conditioning effect. The 1-second gap also affects second order conditioning, but we see reliably more responding to A1 than B1 in this critical probe test. And again, we see an enhancement in this learning. So this suggested that we're again enhancing the learning between A1 and A2 by removing activity of the lateral hypothalamus. So this suggests that the lateral hypothalamus isn't going to come online if one of the cues are motivationally significant, because A1 is still a distal predictor of the outcome. It's still going to oppose learning about it. So this suggested that lateral hypothalamus opposes learning about distal predictors. And I have one more key experiment that I want to show you in particular because I think it's really relevant for cognitive neuroscience in humans and how we think of different psychological disorders. and that is to look at the effect of lateral hypothalamus on latent inhibition. So if lateral hypothalamus is really important for stopping you learning about things that are neutral, distal, it should also be important for stopping learning about things that are actually just irrelevant. And so what we did was to teach our rats that one cue, S1, doesn't mean anything. We taught them that for about 3 days. They had about 40 to 50 pairings of S1 not leading to anything. And then the following day, we take them into the operant chamber and we present S1 as a predictor of food and S2 as a predictor of food. And of course, the basic latent inhibition effect is where they'll learn more quickly in this session about the S2 food relationship than the S1 food relationship. Because S1, they've learned is irrelevant, and so they learn about it more slowly when it suddenly becomes important. And this is disrupted in things like schizophrenia, so it's also a way for us to test whether lateral hypothalamus and its contribution to stopping learning about irrelevant stuff is actually clinically relevant. And so we inactivated lateral hypothalamus during pre-exposure, because if it's important for latent inhibition and stopping attending to S1, we should get rid of latent inhibition when we inactivate it during the pre-exposure phase. And so here you can see our control group, the EYFP group, show a really nice latent inhibition effect across trials in conditioning. So they show faster acquisition of learning about S2 and food relative to S1 and food, so that's the latent inhibition effect. And what we find is we get rid of that when we've inactivated lateral hypothalamus during the pre-exposure stage. So again, this is consistent with an idea that lateral hypothalamus is opposing learning about neutral information, distal information, and also explicitly irrelevant information. And so that's really what's led to our theory that lateral hypothalamus is important for biasing learning towards cues that predict rewards. Because of course we know that it's necessary for learning about those proximal cues. And so thinking about this as a critical arbitrator, I think what we're kind of tapping into here is that we're generally trying to find some sort of balance between learning about things like reward cues that predict things that are important and learning about the general structure of our environment. For example, right now it's coming up to lunch. You might be thinking much less about all the things that are going on and thinking about what you're going to be eating soon or taking a sip of your coffee or something like that, right? If you have a particular need, you're going to be paying more attention and processing more things that are relevant to that need right now. And you'll be kind of tuning out the other stuff that's around you. So this isn't a new idea, obviously. This is a general idea of how we kind of balance our cognitive resources. And I think it's really interesting when you think about it from the perspective of psychopathology. So for example, in schizophrenia, what you see is that people learn more about the general structure of environment and irrelevant information and less about reward predictive cues. And so we've seen this across many decades of research of this disorder, that you tend to get, for example, disruptions of latent inhibition, but also disruptions of learning about cues that predict rewards, especially from Michael Frank's group and things like that. And then if you think about addiction on the other side, it's kind of the other side of the coin here, which is that you see increases in learning about reward cues, and this comes at the expense of learning about the general structure of your environment. So rats that have been exposed to methamphetamine or cocaine or amphetamine actually show disruptions of sensory preconditioning. So I think if we think about this kind of arbitration and lateral hypothalamus being really important for that balance, it becomes really clinically relevant. And so I have a bit of a schematic that we've sort of used that kind of entails the predictions of what we have for how lateral hypothalamus might be changed in these psychological disorders. So here's an example of what a rat's learning and what a human is learning, and this is the idea that you have an optimal bias to rewards. That is, you're still encoding a little bit of what's around you while you're thinking about food and all those things that are going to be upcoming soon, right? So here's a little ratty. He's definitely going to be processing information that's directly related to getting food and reducing his hunger. But he's also going to be processing a little bit of stuff that might be relevant for later on in his environment. Maybe there's some apples buried in that bushel somewhere, right? So it's this kind of balance between all of these things, and this is when lateral hypothalamus is functioning normally. And then in schizophrenia, if we think about lateral hypothalamus as being underactive, and so they're not able to really zoom in on those proximal predictors, instead you have this kind of more general attention and processing towards all information that doesn't have that prioritization in the way that we should. And then if you think about addiction, and we actually know from a number of studies from the physiological standpoint that drugs of abuse will increase activity in hypothalamic circuits, what that would predict, and what you see is the phenotype in people and also in rats that have been exposed to drugs, is that they learn and direct their behavior much more towards reward cues, cues that are proximal to rewards at the expense of other information that is around them. And so, of course, we've got a lot of work to do to actually see if this is relevant at all, but this is kind of our theory moving forward that we're hoping to test formally. And so I just want to show you just one of our most recent experiments that is trying to get at this question of whether activity in lateral hypothalamus might be upregulated following exposure to drugs in a way that's changing the way that we learn about reward cues. So a lot of people have shown that lateral hypothalamus is changed following drugs of abuse, but they haven't tracked this change across learning. So that's what we're really interested in, driving these two features of that literature together. And so this work is led by Ivy Hong, who's a graduate student at UCLA, and we also had a number of other people that contributed in important ways to this project. I'm just going to show one of her new experiments, but Ivy also has a preprint that's currently in review that you can take a look at if you're interested, which shows, you know, all this stuff about the circuit and its relationship. But I'm not going to show that to you today. Instead, I'll give you the TLDR so I can show you the exciting new stuff with activity in lateral hypothalamus. Ivy's essentially established a novel circuit between dopamine neurons in the midbrain and the lateral hypothalamus, and has shown that this circuit is really important for learning about cues and rewards in the way we would expect dopamine to contribute to learning as a prediction error. And self-administration of methamphetamine is producing an enhancement in the control that reward cues have over decision-making. She uses a Pavlovian to instrumental transfer task, so this is something we see in humans as well that have alcohol disorders. And she shows that this is related to a strengthening of the hypothalamic circuit following methamphetamine. So the experiment that I'm going to show you now, which isn't in this preprint, is Ivy's recording of dopamine release in the lateral hypothalamus during cue reward learning before and after methamphetamine self-exposure or self-administration. And I just want to get us into the prediction error mindset. I'm sure everybody has seen this extremely famous dopamine neuron before, and this is essentially the discovery of the prediction error by Wolfram Schultz and colleagues in the 80s and 90s, which culminated in this very famous paper in 1997, where they argued that the dopamine prediction error is a value signal that helps you attach value to cues. And what I want to point out here is the kind of dynamics of dopamine firing during learning. And so we can compare that with what we see in lateral hypothalamus. And so this is recording of dopamine neurons in monkeys, and they're performing a learning task where sometimes they're getting free rewards, that's R here, and when they get a free reward, it's a free juice reward that's delivered into their mouth, you see a phasic increase in the firing of this neuron at the time that reward's received, or 100 milliseconds after. It's very brief. It's like 500 milliseconds between 10 and 20 hertz a second. And it's characteristic across species, which is really amazing, insofar as we can record that from a temporal perspective. And of course, what's really cool about this signal is it backpropagates to a cue that reliably precedes the reward. So once the monkeys learnt that some kind of stimulus in that environment predicts the arrival of food, you now see the phasic increase in dopamine to the cue and no longer to the now expected reward. And dopamine neurons don't just stop encoding what's going on. So if you present the cue that leads to food, and then you don't present the food when it's expected, you see a phasic dip in the firing of dopamine neurons. But what's really cool is the phasic nature of this signal that backpropagates to the cue that predicts reward, right? And so I'll show you what we see in nucleus accumbens, which is the very standard prediction error signal that many other people have shown, right? And so what we're doing here is to record dopamine release in the nucleus accumbens. We use a grab sensor to do that. Grab sensors are great for anyone that's using them. We find that D-light doesn't work as well. But this allows us to record with our fiber photometry signals. So we're getting a bulk dopamine signal from this apparatus. And you can see we're in the nucleus accumbens core if anyone's interested. I don't know what that's called in humans, the core and the shell. Is that the nomenclature that you use in humans?
Yeah, shell.
The shell or the core? Yeah, so we're in the core, so in ventral striatum. And essentially, what we do here is to teach rats that there's one cue that leads to food and one cue that doesn't. And this is their behavior, just over on the right here, how much they go into the food port, and they increase how much they're in the food port for the CS+, we call it, that leads to food, and not the CS-. And this is essentially just the response of bulk dopamine release to those rewards. So you see early in learning in session 1 in black, it's very high to the unexpected rewards, but as they learn that that cue predicts arrival of rewards, it decreases. Everyone kind of ignores that you always still see an increase in dopamine when you deliver unexpected rewards. But you see, we see what happens, which we would expect, is this dopamine signal decreases across learning sessions, as you see in the neurons themselves in VTA, and many people have shown in nucleus accumbens and ventral striatum. And so here we have responding to the cue. You can see that initially there's a little phasic response in black on that first session, but this increases across time as the cue comes to predict food. And you can see this quantified in the area under the curve, where on session 1 you're not seeing much increase of dopamine, but this increases across learning. And we don't see this to the CS-, which never kind of increases and it just stays pretty flat. And so this is just generally what you expect to see in a prediction error signal in dopamine release in ventral striatum, which many people have shown before, and it looks kind of like the prediction error in the neurons, like in Schultz's recordings in substantia nigra and ventral tegmental area. So this is what we were expecting to see in lateral hypothalamus. We should have known better because that's not what we see. But Ivy now is going to record dopamine release in lateral hypothalamus using the same technique, the same system. This is some histology just showing you we're in the lateral hypothalamus. We get expression of GRAB in this very specific part of lateral hypothalamus, and it's also where the dopamine neurons are sending their terminals. So we're pretty sure this is coming from ventral tegmental area, but we're currently doing more studies to establish that very specific connection. And what Ivy's going to do, again, is just to teach them that they have one cue that leads to food and one that doesn't. And this is their behavior, so they increase their food port entries when you present the cue that leads to food and not the cue that leads to nothing. And so here's what dopamine release looks like during presentation of the food reward. So this is actually really surprising because what you can see, the lighter yellow is down here. They're the early sessions of training, and it actually increases with learning. So that's opposite to what we see in nucleus accumbens. We're actually seeing, as the animals have more experience with food, there's an increase in dopamine release at the time of reward. It's also not an error, because it's not a phasic increase. It's a longer, sustained increase across presentation of the reward, which is really interesting, in exactly the same setup as the nucleus accumbens data. And then what we also really love is that we see a ramping of dopamine neurons during presentation of the 10-second cue before the food is delivered. So it looks as though there's an expectation signal that is in the lateral hypothalamus that becomes greater as you become closer to rewards. It looks exactly like a kind of TD sort of expectation signal that you would expect on the basis of the value function. And we don't see this to the CS-. Late learning of the CS- is in black here. It's completely flatlined. What's really interesting is early in learning to the CS-, you actually see this increase in dopamine release in lateral hypothalamus. We thought this was just some weird thing, but we've actually replicated it in other experiments, and so we think it might be an important neural signature that we're interested in investigating. For example, something that's telling them not to learn about that stimulus, which I think is interesting. It's also another reminder to me not to ignore data and not to just say, oh, that's probably a funny thing. And you can quantify these data looking at area under the curves. We've also done waveform analyses that show you in the LH that you increase dopamine release to the CS plus and it decreases to the CS minus, and we increase dopamine release to the reward across time. So this is a really unique profile of dopamine release. And so then what I'll show you now is the studies that Ivy has just done, which is essentially to record dopamine release in the same cue-reward learning task. But first, she's going to give these animals 3 weeks of training with just pressing grain pellets. That's our control. There's no kind of perfect control for these things. This is the one we think is maybe the best, but maybe it's not. We've also done injections of methamphetamine instead of doing self-administration as a way to kind of control for these things, but it is hard. And we compare the dopamine release across learning and controls with the methamphetamine group that have been receiving and working for methamphetamine for weeks, and then we leave them to be abstinent for 3 weeks, so there's no more drugs in their system, and you're looking at the longer-term changes in these circuits. And so recall our kind of prediction is that we should see elevated activity to reward cues in lateral hypothalamus after exposure to drugs, right? The idea is this is upregulated and driving more learning about reward cues. And so here's what our controls look like. You can see again that we're getting that response to the reward deliveries. You can see again that ramping as the rewards are coming up. This actually emerges earlier in training in these rats that have had some exposure to lever presses and all those sorts of things. And then in the meth group, what you can see is we see a significant enhancement in dopamine release to these reward cues and also to these rewards after they've had experience with methamphetamine. And this is selective to the reward cue. We don't see it to the CS minus. So it's not just generalization, it's really this increase in dopamine release to the reward cue. And again, you can quantify this effect across time. We see generally an increase in dopamine. This is our control group in the brown here. Across learning, it emerges a little bit earlier in this group. And you can see that it's significantly elevated in the methamphetamine group. And you also see this elevation across time in dopamine release to the rewards, and it's again elevated in our methamphetamine group. So what this shows is that dopamine release in lateral hypothalamus is increased following methamphetamine self-administration. So again, this is consistent with this idea that lateral hypothalamus is a part of this balance in learning, and following drug exposure, we're seeing heightened activity in this region that might be contributing to this enhancement in learning about reward cues at the expense of other cues. And to kind of pull some of these ideas together, generally, we conceive of dopamine circuits as learning specific things or having particular agendas, right? So lateral hypothalamus dopamine circuits really want to learn about proximal cues, but other areas of the brain, like orbital frontal cortex, we and others have shown is important for learning about neutral cues and sensory preconditioning. And I think what we're seeing is that there's a competition between these agendas that is happening within the brain, and a change in that level of competition or the weighting of those circuits could change the way we're learning about this information. And we have a new paper, or it's 2022 now, where we teamed up with Carrie Bearden and Katie Karlsgott, who are in the clinical schizophrenia field, and we kind of put forward a model of how we might think about the changes in the weights of these circuits contributing to the reinforcement learning deficits we see in schizophrenia. Does anyone have any questions at this point? Tony?
Yeah, thanks, Mel. That's great. I was wondering, that last diagram, in terms of everything you've said around its role in selective attention. So the idea that the relationship to attention and therefore that circuit leading to prelimbic cortex, and that you have direct evidence that in the way it might be regulating attention.
What's sort of interesting is that LH could be regulating selective attention in that way, and I think that is probably the case, but I think it's only going to be for proximal cues and not other cues, because you would have some attentional process that's going on that's helping you to learn A1 and A2 and B1, B2, and understanding how that's different in lateral hypothalamus from prelimbic cortex, I think would be really interesting. We did some experiments with prelimbic cortex and sensory preconditioning to sort of look at just this and whether prelimbic cortex would be important for sensory preconditioning. When you inactivate it, it doesn't have any effects. So it's not producing the same effects as with lateral hypothalamus. So I think there are some differences, but I definitely think we need to think about attention and how lateral hypothalamus is integrated with those circuits, particularly those top-down circuits from frontal cortices. Yeah, I think that's really interesting.
Just have a question.
Yeah.
Generalizable or specific, these two rewards as food rewards versus other rewards, so given the role of lateral hypothalamus.
Yeah, so actually one of our collaborators, Nathan Marchant, today just posted a paper where they've been looking at the GABA neurons and how they're important for learning about cues that are associated with alcohol. And it shows essentially the same thing we showed in 2017, which was in collaboration with Nathan as well. So it looks like it's the same across drugs and reward cues, but definitely would be really interesting to look across other rewards. People have shown lateral hypothalamus is important for social rewards as well, not necessarily the learning about them, that would be the next piece. We also have some data where we've looked at the importance for lateral hypothalamus in learning about cues that predict shock. It's some kind of weird data, but they definitely become important, but things have to happen before it does, but yeah. So I think it is generalizable, although there will be definitely some caveats to that and more work that needs to be done. Yeah. Yeah.
Thank you very much for your time. I want to ask because you presented one of the studies which you mentioned like A1 and A2 and then full association. I was thinking if we increase one more cue like A1, A2, and then we check, it's going to affect.
So if you had an extra step, so you had A1, A2, A3, and then A3 leads to food, I think it would still enhance learning if you inactivate lateral hypothalamus at the beginning because it's a distal predictor. But I think that would be interesting to look at. So would your idea then be that maybe A2 becomes more proximal?
Yeah.
Okay. So if you inactivate during A2 under that circumstance, yeah, that would be interesting. Yeah. We've thought about some things like this, like is it the most distal cue, or is it like, how is it grading temporal proximity? And is it temporal? Can it also be spatial? So I think those are really interesting questions. Yeah. Cool. Okay, so I just have a couple more slides. I won't keep you for too much longer, I promise. And essentially what I just wanted to show you was a new task that we're developing, which is based in human cognitive neuroscience, and we're trying to adapt it for rats in the lab. And that is the door two-step task, and we've made a couple of changes to it. This is led by Masakazu Tara, who's a postdoc at UCID. He came over from UCLA, thank goodness, that was a real savior for me. He does all the computational modeling. He also does the fiber photometry, so it was like, you know, it was going to be hard if he wasn't going to come and, you know, teach everyone else, so that's awesome. And as I mentioned, this is in collaboration with Nathaniel and Marcelo. Marcelo is who we're working with really directly. He's a part of one of our R01 grants, which is transferred over here. Okay, so I think, is everyone familiar with the two-step task here? Not necessarily? Okay, no, that's good. I'll go through it then. Usually when I'm talking to rat people, they're like, what is this? So I never know like the level that I need to go into. So I like to use the example from one of the kid versions because it's more fun. And essentially, Nathaniel's task involves the first stage where participants will see that they need to make a choice between two different stimuli. And that choice that they make between those two stimuli in the first phase will lead to a second stage. So for example, here, what participants, kid participants, are trying to do is to get back home. And there's a particular combination of rocket ships and also these little monster guys that are going to help them get home. And it's always fluctuating which proximal cue here is associated with reward. And one of the critical things as well is that this green rocket, 70% of the time, is going to lead to the red guys, but on 30% of the time, it's going to go to the purple guys by accident. And because the reward contingencies are changing all the time, that can allow us to look at how much the surprising rewards down here modulate how they're going to respond in that first stage. And that tells us whether they're using the specific transitional structure to guess about how they're going to get home. So for example, if they're going to choose a green rocket ship and they go on the common route and they choose this guy who they know that combination is going to get them home, that's all good. And then they're going to choose again green next time and this guy, right? So here they will stay with their choice. They'll make the same green choice and the same red choice that they made before, and that will get them home. However, you can imagine if they have a rare transition, for example, they choose the green, they get the purple guys and they choose this guy and they surprisingly get home because this guy is supposed to help them get home. Now, what they do on the next trial should be to shift. So they shouldn't stay, and instead, when they get to the first stage, they should choose the blue rocket, because even though last time it was the green rocket that led to these purple guys, actually you're more likely to get to the purple guys with the blue rocket. Does that make sense? And this shows you that people, or rats, are encoding the transitional structure that got them to this point, rather than just relying on what we call a model-free network, where they would just be always repeating the choice that was rewarded. How is that? Does that make sense? Yeah? Cool. You guys are easy. So essentially what we're trying to do is to leverage this task to be able to formally test with computational modeling how lateral hypothalamus and the wider circuit arbitrates between learning about the first stage distal predictors and the second stage proximal predictors. And because of course this is developed for computational learning, we can have thousands of trials where we can look at the impact of computational modeling, thousands of trials where we can look at the impact of changing these circuits in the way that they behave. Rather than a lot of the one-shot tests we often do between A1 and B1, and it tends to be quite variable, and it doesn't lend to modeling in the same way. So here's our little task. So the rats first tell us that they're ready to start a new trial, and they stick their nose in the food port, and that will lead to the presentation of 2 distal cues. So this is like stage 1, and it will be either a click or a white noise. And after those 5-second cues, they're presented with our choice, and they're going to choose either the left or right lever. We have some forced choice trials and some free trials. We just analyze the free trials. And then what happens is we come up to this probabilistic state transition where we have common in blue contingencies and also those rare contingencies. We do this on the basis of 80-20 rather than 70-30 because the rats are better at doing that. And then they have these proximal cues. So we have a tone and siren, and they're predicted differentially by a combination of the distal cue and the choice of left or right lever. And one of those rewards for part of the session, one of those cues, will lead to reward and the others don't. We do this probabilistically, but it's about 90-10. It's not in the same way as they do with humans. And so the idea is for some of the session, the tone's the one that they want to find, right? Because it's getting rewarded. But this could change at any moment because it's fluctuating throughout the session. And so you can imagine if a rat initiates the trial port, it gets presentation of a click, so it goes left because it's trying to find the tone because that's what's being rewarded at this point. But it unexpectedly goes to the siren, and it unexpectedly gets a reward, just like in the first example with the rockets. On the next trial, they shouldn't stay, and instead they should shift if they get this click and choose the right lever to be more likely to get that siren presentation. Does that make sense? So it allows us to test in the same kind of way whether animals are using that transitional structure to regulate their behavior. Importantly, the difference between our task, we have these Pavlovian cues, so the cue presentations as separate from the instrumental behavior, because that will allow us to inactivate the circuit during those cues without impacting behavior, which is important. And so I'll just show you some of the basic data. So we're still at a point where we're trying to develop this task. We haven't done any of the neuroscience yet. And what you can see is that this is the specific, sorry, that's come up wrong, so you can see what I'm about to show you. But this is the schematic of the task, for example, where the pink tone is going to be leading to reward, but this is shifting across the session. And you can see in the proportion of correct that rats are pretty good at this. They're pretty good at making the right choices, right? So their stay probabilities are being modulated by the transition structure and whether or not they were rewarded. And here's what data should look like. This is an example from some of Nathaniel's data in humans. You see this is looking at stay probability. So if you're not thinking about the transitional structure of the task, essentially you should just stay with what's ever rewarded and change your response on the next trial if you weren't rewarded, right? But if you're taking into account how you got there and whether it was a common or rare transition, you should modulate your stay probabilities on the basis of those transitions. So for example, if it's a common reward, you chose the green, you went to the red guys, right? And you got home, you should stay. But if it was a rare reward, so you went to the purple guys and you got home, now you should switch your response. And this is essentially just capturing the way in which you should change your behavior and your stay probabilities. And so this is what our ratties are looking like right now. We've actually got it looking a little bit better as they kind of move through the different training structures. And you can see most critically, we have this really nice difference between whether they stay or switch based on whether the reward was determined by a common or rare transition. We don't see this as much with the unrewarded trials. Rats don't seem to be as sensitive about the non-rewarded trials. They tend to stay. Other people have shown this in different versions of the two-step rodent task, but we are actually getting them to be a little bit better now. We're also looking at logistic regressions where we can take into account the coefficients of how they're influenced by reward or transition structure, and we're starting to see that they are showing a difference in their stay probability on the basis of whether or not they were rewarded, and also on the basis of which transition it was. So we're still trying to improve this task. The ratties are still running. But we think we're really getting somewhere, which we think is going to be essentially a powerful tool to try to understand from a formal perspective whether lateral hypothalamus is doing what we think it does. And of course, people with various psychological disorders show differences in the two-step task in humans, so we think it's a way to try to make a connection with the human cognitive